Ladies and gentlemen, I've been informed that the next witness is Nicole Graham, who will be testifying by video. During her testimony, she will be making reference to Exhibit 15 in her previous testimony. For purposes of this hearing, it's Exhibit 22. She'll also make reference to Exhibit 119, which for purposes of this trial is Exhibit 47. And Brittany Ziem, who testified earlier, made reference to Exhibit 106. That's now 2. She also made reference to Exhibit 6, which is now 14. Now, if that's not confusing enough for you all, it's confusing enough for me, but the lawyers thought it was important that I tell you all that gobbledygook, so I did. Okay? Call your next witness.
Thank you, Your Honor. The people, we call Nicole Graham. By stipulation, Ms. Graham's testimony from the last trial in March of 2021 will be played. So if I may play that, Your Honor.
You may. You can have it.
Good afternoon, Ms. Graham.
Good afternoon.
Could you please spell your full name for the record?
Sure. My name is Nicole Graham, and ICOLEGRAHAM.
And how are you employed?
I'm employed with the Michigan State Police at the Lansing Forensics Laboratory. I'm a forensic scientist and also a unit supervisor in the biology and DNA section.
And do you have any specific educational background that enables you to perform DNA testing?
I do have a Bachelor of Science degree in biochemistry from Alma College, and I've taken some graduate level coursework in forensic DNA.
And you have experience, or how much experience do you have as far as DNA analysis?
I've been a forensic scientist for 23 years now. I started out with the Florida Department of Law Enforcement, where I worked for 2 years, and then worked for 8 years with the Virginia Department of Forensic Science, and I've been with the MSP for over 13 years now.
And are you proficiency tested?
Yes, twice a year.
Now, do you know the number of times or approximate number of times that you've been qualified as a DNA biology expert?
I do keep track, and I checked yesterday, and it was 119 times prior to today, and that's in Florida, Virginia, Michigan, as well as federal courts.
And with that, Your Honor, I move to qualify Ms. Graham as an expert in forensic DNA analysis.
Mr. O'Farrell.
I have no questions, Your Honor, and I would stipulate.
She will be so recognized.
Thank you, Your Honor. First, if you can explain to the jury, what is DNA?
DNA is a biological molecule that's found in most of the cells of your body. It is the genetic material that you inherit, half from your mother and half from your father. And DNA does code for things like what color eyes you have and how tall you become. But a lot of DNA is the same between all humans. It codes for the fact that we have 2 arms and 2 legs. There are some regions that differ between humans, and those are the areas that forensic scientists look at in order to differentiate between biological material from different people.
And now, can a DNA sample be obtained from an object that someone touches with their hands?
Yes, years ago when forensic DNA first came around, you needed a blood stain or a semen stain that was kind of large. This was back in the late 80s and early 90s. Over the years, it's gotten far more sensitive. So for the last 15, 20 years, probably we started noticing we could get DNA from touch DNA or contact, just an item that somebody handled or held or touched.
And is there anything more, I guess you could say, about how the DNA would transfer from the skin to the object?
Yeah, it's actually, with COVID, it's almost better, I think easier for people to understand. We're all wearing masks. We've been wearing masks in the DNA laboratory for many, many years. Those tiny micro droplets of saliva that we think could carry viruses that could land on the surface or you could inhale, all of those contain human DNA from the person that it comes from also. So DNA can be spread by talking, coughing, it sheds skin cells like you scratch your head or have some dandruff that could have DNA, or just sweaty hands, biting your nails. Any of those could be DNA on surfaces that you touch, or even if you don't touch it, if you're just talking near it, some of your DNA could land on those items.
Now, when law enforcement or someone from your office wants to collect DNA from an object, what exactly will they do?
Most touch DNA samples are collected through swabbing. Usually it's just a regular cotton tip swab, like a Q-tip, and they just moisten it with a little bit of water and then just rub the surface and try to collect as much of that area as they can and send it to the laboratory.
All right. And then as part of your analysis, you'll compare that with a known sample from a human, is that correct?
Yes, in general, what we do is develop a profile from an evidentiary sample, an unknown, and then we also develop known DNA profiles from reference samples that are taken directly from a person, and then we can compare and see if they're the same or different.
And how would a sample be collected from a human?
It used to be blood samples, but that's pretty invasive. So normally nowadays we just get a buccal swab, which is kind of like a saliva sample, but they'll actually rub inside the cheek and try to collect some of those cells from the inside lining of the cheek. We can still use blood too, though.
Now, just going to ask you some questions about the testing procedure. Now, obviously, you're able to compare a sample obtained from whatever object and then compare it to a person's known human sample, correct?
Correct.
All right. Now, and then I'll first ask, does your testing follow standardized written procedures?
Yes, it does. We have a very extensive, detailed protocol that we must follow.
Okay. Now, so you have a swab where you've collected the DNA sample from either some object or a person. How do you get that DNA off of the swab so that you can analyze it?
Quickly, they just cut the swab and put it into a little tube. And then we add a series of chemicals that break open the cells and release the DNA. We perform some steps that purify that DNA and remove protein and other cellular debris so that we have just DNA. We do a quick quantification step that tells us, one, if it's human DNA and how much of it that we have. We then do a procedure called polymerase chain reaction, or PCR. And this is where we get our sensitivity from. It actually targets, in our case, we target 23 different regions on the human genome to copy many times over. It just mimics the same process your body does when it creates new DNA to make new cells. We do that in a tube. Once we've done the PCR and we have more DNA, it goes into an instrument called a genetic analyzer, where the DNA fragments are separated by size. The output of that is what we call an electropherogram, and it's basically a series of peaks. It'll kind of be a baseline, and then there'll be a peak wherever it sees a DNA fragment. And those peaks across the 23 different regions we're looking at form a pattern that we would term a DNA profile. That DNA profile can then be compared to DNA profile of the known reference sample to see if they're the same or they differ.
Now, from that profile, can you determine how many contributors there were to that DNA sample?
Yeah, sometimes we get single source samples that would usually be from something like blood, where it's just coming from one person, and those are very easy to interpret. But a lot of the times nowadays, especially when you're dealing with contact DNA, we're getting a mixture where we have multiple contributors, multiple people's DNA mixed in there together. Because you inherit half of your DNA from your mom and half from your dad, you generally have two types at each of those 23 regions. You see two peaks. Occasionally, your mom and dad happen to just give you the same type, so you'll have just one peak, but it'll be twice as big. Anyway, so a single source sample, you don't expect to see more than two peaks at each region. Once you start seeing 3 and 4, oh, I have at least 2 people in here. Once you start seeing 5 and 6, oh, I must have at least 3 people in here. And if you get 7 and 8, now you're up to 4 contributors. Now in our laboratory, we do not interpret anything more than 4 contributors. So once we see a 9th or 10th allele indicating a 5th donor, then we say that it's too complex to interpret.
And then is this interpretation or analysis as far as how many contributors there are, can that be done visually?
It is done visually, yeah.
Okay. And as far as comparing them, and then the next step is you would move on to compare the profile to a known sample. Is that correct?
That's correct.
All right. Now, your lab wouldn't do that if there's more than 3 contributors. Is that right?
More than 4.
More than 4. Okay. So what is used to do the comparison between the known and the DNA sample from that object?
If it's single source, we can visually just look at them and see if the types are the same or different. And if they're different, then it's an exclusion. It's not that person. And if they're the same, then we have to assign a statistical probability to the weight of that match. This stems back from early on when they did ABO typing. You could say, oh, the blood match, they're both type O, but that's 45% of the population. So it's not very discriminating. So we're required in Michigan by law to provide a statistic that goes with that match. Now, with a mixture, the process is similar, but it can get a lot more difficult. So we have a new software called StarMix. It's called STRMIX. And it actually performs what we call a mixture deconvolution, where it can kind of try to separate out the different contributors, what the most likely types are from the different contributors to a mixture. And this can help us with our comparison. The other thing that StarMix does for us is calculate a statistic to go with that, either match or inclusion of a profile.
And do you have a term you use for that statistic?
The statistic that we currently report using StarMix is known as a likelihood ratio. It is, you would compare 2 different hypotheses. So the first hypothesis would be that the person of interest that you're comparing is a contributor. And then that's weighted against the hypothesis that they're not a contributor, it's someone else. The likelihood ratio, if it's a 0, that means they're excluded and they are not a contributor, they're outright excluded. If you get a likelihood ratio of 1, that means there's equal probability between the 2 hypotheses. It's just as likely they're in there as not. So that's basically like an inconclusive. We can't really tell. If you get a likelihood ratio greater than 1, that indicates support for the first hypothesis, that that person of interest is a contributor to that mixture. And the higher the number that goes, the more support that it gives to that. So to say a likelihood ratio of 100 is not very strong support that they're in there. And if you go up to 1 million or 1 trillion or a quintillion, that strength goes up. Now you can also get likelihood ratios that are less than 1, and those would lend support to hypotheses 2 being more likely to be correct. That they're not in there, it's someone other than them. They're not outright excluded, but it doesn't really look like they're in there. We don't get those quite as often, but that's another possibility.
Now, with respect to that StarMix software, is that something that's generally accepted in the scientific community?
Yes, it's gaining a lot of popularity. It's still fairly new. It was developed in 2012 in New Zealand. It's been used extensively in Australia and New Zealand since then. It then moved into America, Canada, and Europe. And I checked their website just the other day, and they said they've just had their 220,000th case using StarMix since 2012 worldwide. So it's become very popular. There's 59 labs in the US that are currently using it on casework, but there's another 50 to 60 that are in the process of validating it to go online.
Okay. Thank you, Ms. Graham. Now, did you receive any samples in this particular case?
I actually analyzed the data. The laboratory did receive samples.
And were there samples that the lab actually received related to this case?
Yes, there were.
And what were those?
There was 2, one evidentiary sample, which was swabs of a laser site, and the other was a known from Davion Thompson.
Okay. And who were the samples received from, or where were the samples received from?
The samples were submitted from Saginaw PD to our Bridgeport laboratory. A scientist there actually swabbed the laser site, she actually received the item and swabbed it in the laboratory and then forwarded that swab to the Lansing lab for DNA.
Okay. And someone else in the lab then does the extraction, runs it through the genetic analyzer, and then you receive the data. Is that correct?
Yes, in this case, we are all trained to do the lab work and the interpretation, but sometimes cases can switch hands. It's kind of a matter of efficiency. So I'm trained to do the same things, but I did not perform the lab work in this particular case. I just received the data and did the interpretation and the star mix.
Okay. So all the testing steps, the testing procedure you just outlined, that was all followed in this case?
Yes, we're all required and we're all proficiency tested, but we're all required to follow those same written procedures.
Okay. Now, when you took a look at the data, were you able to determine whether the DNA sample from the gun site is suitable for comparison with Mr. Thompson's sample?
Yes, it was suitable.
And were you able to determine the number of contributors?
Yes, in this case, there were 6 alleles at a couple of the locations. So that indicates to me there's at least, that there's probably 3 contributors, and there was no indication that it was more than 3. In this case, there was sort of a major person, like a high level contributor, and then 2 very low level that were only partial. I didn't see their types at all of the regions.
Now, I'll get to the stir mix analysis in a minute, but with the profile that you saw, was that something that you could visually analyze yourself?
I would be able to, yeah. The major donor, it's our policy to just use star mix no matter what, but the major was obvious to me visually also.
Okay. And then you then ran the data that you ran that into the star mix program, is that right? Okay. And were you able to reach a conclusion or get a result as far as the star mix analysis?
I was. So star mix did the deconvolution and it gives you a mixture proportion. So it said that contributor 1 was about 93% of the mixture, and then contributor 2 was about 5%, and contributor 3 about 2%. So you can see that lined up with my visual interpretation that I had 1 major contributor and 2 very minor. It then went ahead and what it does is it pulls out what it thinks the most likely explanation is for each of those contributors. So for contributor 1, it was able to pull out a complete profile and say, I think this is contributor 1's complete profile. I then ran it against Mr. Thompson's DNA, known reference sample that we had also developed a profile from, and it lines up with that contributor 1 profile. So it gave a very high likelihood ratio because it fit perfectly with that first contributor to the mixture. The likelihood ratio that we got in this was 1 octillion. An octillion is 27 zeros. Anyone doesn't know that.
Okay, and so just to clarify, so the Thompson sample was the 93% contributor, is that correct?
Correct.
And so what's that, can you just state the likelihood ratio in, I guess, in total as far as what that 1 octillion means?
Yes, so my actual conclusion was, hypothesis 1 was that the mixture was a combination of Davion Thompson and 2 unknown individuals. Hypothesis 2 was that it was 3 unknown individuals. And then that likelihood ratio was that it's 1 octillion times more likely that it's Davion Thompson and 2 unknown individuals than 3 unknown individuals.
And from that, are you able to conclude as far as the strength of the support that the defendant is a contributor to the DNA profile?
Yes, we include a verbal equivalent scale in our report that gives numbers of limited support, moderate support, strong support, or very strong support. That's because this likelihood ratio is a little tricky. It's a unitless number. There's no unit to it that I can say what is a big number really. So I think in this report, it was over 10,000 we called very strong support. We've actually just recently, since this report's been written, we've increased that scale. We've gone a little more conservative, and now we say anything over 1 million is very strong support. So obviously in this case, an octillion is more than 10,000 or 1 million. So it falls into the very strong support category pretty solidly.
And as far as, so you explained the strength of the results with respect to Mr. Thompson. So just to clarify, as far as determining, so hypothetically, if you have one contributor who had actually touched an object, these results wouldn't tell you the actual time, like when that contributor touched an object, correct?
That's correct. Touch DNA is a little bit tricky because it can tell you pretty well who someone is, but not when the DNA got there or how it got there. So it's very stable for years. If an object is just left alone and it's not really further handled, your DNA can stay on there indefinitely. And it can vary. You could touch something and not leave your DNA. You could not touch something and possibly get your DNA on it through transfer. So it's kind of tricky to say anything definitively of how it got there.
Okay. So as far as the minor, would you call the other two contributors, 2 and 3, would you call them minor contributors?
I would.
All right. What could you say about those contributors as far as, possible scenarios of when they could have touched the gun site?
Well, the same kind of thing. So there are 2 other people on the evidence. There's no way to say for sure that the last person that touched it is the major. That's often the case, but that's not always the case. I researched a lot of articles preparing for this, and unfortunately, they're trying to do more and more studies on touch DNA and who leaves it and when, and there's just a lot of variables. So the example I like to give is if you just went to the bathroom, washed your hands really thoroughly, and you put hand sanitizer on, and you come in through that door and you open the doorknob, you're very unlikely to leave any DNA. Your hands are clean. But I always use the example, say you're a nail biter, like I am sometimes, and you're chewing on your nails and you're leaving saliva, which is a highly concentrated source of DNA, and you grab that door handle, you're quite likely to leave DNA. So it really just depends on, it also depends on the surface you're leaving it on. Textured surfaces, porous surfaces, they tend to hold on to DNA better than a smooth surface. So something like glass doesn't do well. This table's pretty smooth. I wouldn't probably easily leave a lot of DNA, but we get really good results off gun grips or screwdriver handles where somebody was hanging onto it tightly, and with some friction rubbing on a textured area, it will leave behind a lot of DNA. So there's just a lot of factors that vary. And then there are some studies that kind of hypothesize that some people just seem to shed more DNA than others. And they don't know if that's a permanent condition, like if someone's just always a DNA shedder, or if it's temporary. Again, it might be because one's a nail biter and one's not or something, but it does tend to vary between individuals on how much DNA they leave on things.
Okay. And is there anything you can say about potentially, talking about a gun site, Exhibit 15 is on the screen. I believe you've seen that before.
Yes, I've seen a picture.
Okay. And with respect to that, I mean, what people hypothetically could have touched that?
Well, I would say I didn't actually see the item or swab it. One of our scientists did. She has in her notes, and I reviewed them before I came here, that she swabbed the textured area on what would be the right side of that photograph. This is generally what we do on these kind of items for 2 reasons. The textured areas are where we get the most DNA from, and conversely, the texture areas are very bad for fingerprints. They never are able to get usable fingerprints off those rough textured areas where they like the smooth surfaces. And smooth surfaces don't work well for DNA, but work well for latent prints. So this item was going, I believe, to latent fingerprints after DNA. So she swabbed the textured areas.
Okay, so and let's kind of try and get a little bit more specific. So let's just assume there's contributor one and that person had actually handled the object. The other two minor contributors at 3 and 2%. Do you have any examples of who reasonably could have handled the laser site?
Oh, well, so DNA is just everywhere. Most things are not sterilized. We've even found that there can be DNA from manufacturing plants. They don't actually sterilize items before they put them on store shelves. We've had actually some contamination of supplies from employees of the labs that make store supplies. So it could be from that, or it could be from somebody talking near something. If you weren't wearing it, actually probably going to be better in 2020 and going forward. People are wearing masks, but any other time if somebody's coughing, sneezing, talking near an item, just tiny droplets land on that. It could be from someone who used it previously. It could be from someone who used it most recently and they just didn't leave a lot of DNA. Their hands are really clean. It really could be anyone that is. At any of those three contributors, there are multiple explanations for how they got there.
And then as far as what would be your opinion as far as that number one contributor, that 93% contributor, if you couldn't make one?
Well, it is a fairly low level sample, but not super low. We actually go quite a bit lower. And they are a nice clear major at 93%. So they are more likely to have made direct contact with it, but it's not 100% conclusive. So you are more likely, I'm more likely if I touch this table to lose my DNA than if I shake your hand and then you touch the table for you to lose my DNA. It can happen though, but it's less likely the further you get out. So
now maybe just explain a bit of kind of going into that secondary transfer. What is secondary transfer?
So primary transfer is direct contact. It means my DNA is on it because I touched the item, I handled it, I was holding it, or even, I was right here and I talked over it and my DNA is on it. It's direct close contact. Secondary transfer would be when some other vector was used to transfer your DNA to an item that you didn't have direct contact with. Years and years ago, when we had, a blood stain the size of a quarter, they said, secondary transfer is impossible. That can never happen. We would have testified to that 20, 25 years ago. But as the testing got more sensitive, it became possible. So that's kind of the double-edged sword to the high sensitivity we have now with the contact DNA. We can get DNA we didn't detect before, but now there is a possibility of secondary, even tertiary transfer. But obviously, as you get less direct, it becomes less likely.
Okay, so there could be contact between one person, person A, they touch person B touches the object. There is a possibility that person A can get, person A's DNA can be found on that object. Is that reasonable?
That's correct.
Okay. And is there anything you can say about that? Any studies you've seen as far as any additional information on that?
Well, it's hard to quantify, and that's kind of hard for us as DNA people. We've always been very proud of being able to give that statistic. I give you that likelihood ratio of an octillion, but that is only the likelihood ratio of his DNA being in that mixture. It has nothing to do with a likelihood of him touching it or having used it or committed a crime or any of that. Those would take additional information that we don't have. So most of these studies I read talk about how we need to do more studies, and they're talking about building data banks of statistics for how often you transfer your DNA and how often when you touch this item and lose this much DNA. We're a long ways from that. So I don't have any hard or fast number that I can get to the likelihood. I reviewed a number of papers and several that summarized other papers. All of them agreed that it's more likely to have primary transfer than secondary transfer. Obviously, secondary transfer is more common than tertiary transfer. It kind of varied. There was one study they were saying that they washed their hands, then they put on gloves for like 2 hours, so they get nice and sweaty in there. And then they made them shake hands for 2 minutes, which is kind of unrealistic. I don't know anyone shake hands vigorously for 2 minutes. And they said about 25% of the time in that study, they detected only the person who didn't actually touch the item. About 75%, they got a mixture or just the person who actually directly touched the item. Then another study, they did a similar thing, but they did 10 seconds of hands shaking. And in that case, they didn't detect anyone. Or they detected secondary transfer, but he was always the very minor donor. There was another part that they said they summarized like 6 different studies that said that in total in 6 studies, less than 3% of the time was secondary transfer the major contributor to the mixture. So it's not impossible, but it is less likely.
Now, when you do get in that example, so person A to person B, with respect to person B's DNA, is that going to get on the object too? Is that kind of reasonable to assume?
Usually, so that'd be the most common. Is if I shake your hand and you transfer my DNA, you're transferring your DNA with it. And most of those studies said usually they had more DNA from the direct transfer person than the indirect transfer, but not 100% of the time. There were incidences where they only detected the indirect person and not the person who did the transferring. And again, that could have to do with, say that the intermediary person has really clean hands, he just washed them, and person A has just been biting their nails and leaving more DNA. So it is possible.
Okay, and so let me kind of ask you about, so an officer who is handling the evidence, what are some possibilities as far as, even though they're taking the precautions necessary, that their DNA could get on and on?
We try to train officers. We have people that go to crime scenes. The biggest thing is probably should be wearing a mask, or at least not talking over the evidence, because you can move your DNA that way, and wearing gloves. And they're very good at wearing gloves, but I don't know that all of them maybe change their gloves between every item they touch. In the lab, we are really, we know how sensitive it is. We change gloves just constantly. So I think one of the biggest risks is they're wearing their gloves. They're worried about, they don't want to get their DNA on the item. But what if they touched something bloody from the victim and now they touched a different item? They can be transferring some of that blood DNA onto the other item.
Okay, and then I think you mentioned wearing masks. Officers wearing masks when they're collecting evidence.
Yeah, like I said, that's been our requirement. As much as they're annoying and they clog up their glasses and everything, we've been required to do that in the DNA lab for a very long time, because we realized how much of a source of contamination is even breathing. I mean, honestly, it's not even just talking. You can be careful not to do that. Sometimes you can't control a cough or a sneeze, and even sometimes just breathing.
Okay. So back in 2018, I mean, now officers are all wearing masks, right? Back in 2018, it wasn't as common when they were collecting.
Probably not. No.
Okay. And so an officer talking, saliva could get on the object, as you're saying, and so the DNA from the officer could get on it.
Yes, I mean that any other DNA on the item could be, like I said, from the manufacturer, from anyone who's ever used it, been near it, handled it in the interim, from the officer at collection. Even in the laboratory, we take extensive precautions in the laboratory to avoid contamination, but it still doesn't mean it's completely impossible. We're still humans and we have DNA, so.
Okay, and then someone, obviously, the gun site, it's going to be sold at some point to some person. Is it possible that the person who sold it, assuming it wasn't packaged or something like that, that could be someone who gets the DNA on?
They could come in a box, but yeah, I don't know. Anyone who's come in contact with it, however long it's been in existence, could have left their DNA. And like I said, it can last. It's very stable molecule. It'll last indefinitely on the surface if it's not subjected to high heat, humidity, direct sunlight. It's not wiped away. It will stay on there.
Okay. So basically, this kind of highlights that we only have 3 actual contributors on this particular item, where, when there's so many people that could have come in contact with it. So is that significant?
It's really nice, but we actually kind of officially refer to these as nice mixtures when you have only 3 and it's a clear major like that, because we get a lot of 4-person mixtures that maybe a 25 to 25 to 25 to 25, and those get tricky because you can't tell which ones pair together because everything's the same height. This wasn't low level. I wasn't seeing extensive dropout. It was a nice mixture. As far as could there have been other people's DNA on it over the years? Yeah, and it could have been removed. That's another thing that can happen with touch DNA is that when you're touching something and leaving your DNA, you can also be removing the DNA from someone who touched it previously.
And now, and just to be clear, and I think you have been clear, just because someone's DNA or someone's DNA is consistent with being on the object, that's not 100%. In your Sturmax analysis, you have to consider other things with that to corroborate the fact that that person actually handled it, correct?
Right. I mean, the Sturmax is not making any judgment onto how anything got there, how long it's been there. It's only looking at the probability of that DNA profile existing, giving this person as a contributor versus not. So that's why, like I said, it gets tricky testifying to these because I can't give a similar likelihood ratio to say the likelihood of, okay, it looks very strong that his DNA is on there, but how did it get there? I don't have any way to give a firm number to that.
Okay. And with all that in, let me ask you just to kind of close. So considering the likelihood in this case, is that likelihood ratio in this case consistent with an argument that Mr. Thompson never touched the laser site shown up on the screen? Is the likely ratio consistent with the theory that Thompson never touched it?
Well, the likelihood ratio is very strong for that it's his DNA on the gun. Again, it doesn't really factor in the likelihood of them touching it. Now, from my experience and the articles I was reading research, the most likely explanation of his DNA being on there would be because he handled it directly. It's not the only explanation, but it would be the most common or most likely explanation to be a major donor on it.
Okay. Thank you, Dr. Ms. Graham.
Mr. O'Farrell, I don't want to put you on the spot. How long do you think you'll be? We can take a break. You're going to be longer than 15 minutes. And if you are, that's fine. We'll take a break. I need a break. I'm sure other people may need one as well. We'll take a short recess. Let me go to my clerk, please. Please be seated. Mr. Fedoruk, I understand you have not concluded with the witness yet. You may continue.
Thank you, Your Honor. Just briefly, Ms. Graham, I forgot that you did prepare a report pursuant to your analysis in this case, correct?
That's correct.
Okay. I'm gonna show you that. It's marked People's Exhibit 119 for identification. I'll show you that in a minute. Is that what you do when you do an analysis, DNA analysis? Is that typically what you do is prepare a report?
Yes, we're required to prepare a report on all of our analyses.
Okay, and in this particular report, does your lab report, does that reflect the steps that you took in the analysis and the conclusions that you drew?
It includes the general process and then the conclusions, including statistics.
Okay, and does it reflect your testimony?
Yeah.
Okay. So, Ms. Graham, after reviewing People's Exhibit 119 for identification, is that the report that you prepared for the case?
Yes, this is a copy of the report.
People offering to evidence 119, People's 119.
Mr. O'Farrell,
No objection.
Exhibit 119 will be admitted. Mr. Fedoruk, I take it you're through since you're sitting down.
I apologize. That's all I have.
Cross-examination, Mr.
Good afternoon, Ms. Graham.
Good afternoon.
The verbal scale on your report, and I'm looking at page 2, indicates this analysis provides a very strong support that Davion Thompson is a contributor to the DNA profile developed for the swabs of laser site, correct?
Correct.
So being a contributor, then we need to determine all of the nuances that you've been describing to the jury about how that DNA made its contribution.
Yeah, we'd like to, but it's almost impossible to really say for sure how it got there or when.
Exactly, okay. So you referred to other members of your team doing a part of this investigation. Who would have developed the sample that was obtained from the site?
Katie Urka, who's a scientist in our lab, I'm her supervisor, actually did the DNA, the laboratory analysis and developed the DNA profiles that I interpreted.
Is there any quantitative value made of the size of the sample that was obtained?
Yes, that's one of our steps after we extract the DNA, is to quantify how much human DNA is present.
Would that have been part of your role or part of her role?
That was part of her role. But as when I took it over, I received all of her data worksheets, so I have access to all of that in my interpretation.
Okay. And after you had provided the report in this case, and I obtained a copy, I had contacted you requesting the worksheets and other data. Is that correct?
Correct.
You were kind enough to email that to me?
Yes.
And would your worksheets be available to you so that you could give us an idea of the quantity of the sample in this case?
Yes, I have a copy of the quantitation results. Is it all right if I refer to them?
Yes.
Could you just tell us what the result was as far as the total DNA sample?
Sure. For the sample on the laser site, it was 0.01227 nanograms per microliter is the concentration. So a nanogram, just for reference, if you know what a milligram is from like medicine and stuff, roughly an idea, it's pretty small. A microgram is 1,000th of that. And then a nanogram is 1,000th of that. So it's a very, very small amount.
And then the other, you concluded there were 2 other contributors?
That would be the total DNA in the whole sample, but that would be divided amongst the 3 contributors. Yeah.
3 including Mr. Thompson, or a total of 4?
Oh, total of 3.
Okay. And that sample then is the total human DNA of all contributors, right?
Correct. And that was the concentration. So then we put 15 microliters of that into the amplification reaction. So I amped a total of 0.18 nanograms of DNA. Generally, we would target about 0.5 nanograms. So it's a little less than half of what we ideally would want to put into the PCR reaction.
And you had mentioned that part of this process, once you had the known sample of Mr. Thompson, would be quantitatively determining his contribution.
Well,
I'm sorry, go ahead.
you know, and I've been doing this for a long time. So it used to be visually, I would declare a major donor. So when I first visually looked at it, I'm like, I see a major donor. But we do by policy now run it through StarMix and let it come up with those mixture proportions that I was talking about, that broke it down and said it was about a 93 to 5 to 2 as the 3 contributors.
Okay, so StarMix is a software program, which I guess supplements your own conclusions. It doesn't replace them. Is that correct?
That's correct. The primary diagnostics on the StarMix run is if it's intuitive. If now I look at it and that makes sense to me. If it had come back and said this is a 40 to 20 mixture, I would have said no, that's not at all what it is. But 93 to 5 to 2 was about what I would have visually guessed.
You talked about the proclivity of DNA to attach itself to things, and it can happen in almost as many ways as you can imagine. Is that a fair statement?
Yeah, this is almost infinite number of possibilities for transferring and removing and redepositing
And is that complicated, or at least made more difficult because when the sample is smaller?
yes. I mean, that's a good point. So DNA can be transferred when it's a larger amount, but it's less likely that you're going to do that without noticing. A blood sample, if you touch liquid blood, you could easily transfer it to something. You're going to see it. So when you get the small amount of DNA, especially contact, it's invisible to the naked eye. You can't really trace where it's coming from or going to.
So I could touch an object, and then is it my skin cells that do the DNA, or what exactly is it about me that then shows up as DNA on the object?
And that's the interesting thing. They're still kind of trying to research that too. They're not sure. It's probably a variety of sources. Anyone who touches their face, there's a lot of tears, nasal secretions, saliva are all high sources of DNA. If you're scratching your skin, you could be removing some skin tissue that way. They do believe that there's sweat that can kind of carry it out of your pores, but there may even be some free DNA in your outer layer of skin. Because remember, your outer layer of skin is kind of dying off, it's constantly renewing itself from the inside. So the outer layer, sometimes there's just DNA that's from old dead skin cells that have gone away. So it's kind of a combination, and they're not real sure. And that's why they say some people may be more prone to leaving their DNA than others, but they're not sure what factors. They've actually found that it seems to be more in men than women, but they don't know why.
And then my DNA can get on an object that I've never even touched, correct?
It's possible, yeah.
And is that where we get into the concept of transfer DNA? Is that the term that's used?
Yes, well, you can have, it's still considered direct or primary transfer if you say you didn't touch something, but you were coughing over it and your saliva was landing on it. But it can also be transferred there through an intermediary, usually another person, but also it could be from an object. You've got a sweatshirt you wear every day and you never wash it and it has a lot of your DNA on it. That could possibly contaminate another object.
Is it conceivable that if I had your sweater and I wanted to place your DNA on an object, I might rub your sweater on that object and then that would show your DNA on it?
It could happen, it could be possible. Again, it would depend on how clean the sweater is. Had I just washed it, or again, it's my favorite and I wear it and I never washed it. That's the thing, almost any scenario is possible.
Okay. And there's really no way to quantify the statistical probability of all of those, any of those scenarios. Is that correct?
No, not at the current time. I just read an article in Europe. I think they're trying to do that in someday, but it sounds like kind of a tremendous undertaking to try to develop probabilities for every possible DNA transfer.
So I understand correctly, what we can say about that gun site is that it had DNA contributions from 3 different people.
There were 3 people in it.
And the most highest concentration was from Mr. Thompson.
Correct.
So we can't make any comparisons about those 3 as to who actually touched the object.
Right, I can't. I mean, that's the most likely scenario for getting your DNA is if you had direct contact, but it's not the only.
Okay. And if a person, say the lesser one, had access to an item of Mr. Thompson's clothing or whatever, they might contribute his DNA just as a way of establishing a false probability. Is that right?
Oh, intentionally? I mean, I suppose that's possible. Or unintentionally.
Sure. Did you form a conclusion as to whether these three contributors were male or female or a combination?
So the major donor, which was consistent with Mr. Thompson, was male. The problem with that is we just see that there's an X and a Y chromosome, but when you have those minor donors, so everybody, male and female, would have an X, and only males would have a Y. Once I see that big Y in the major donor, I can't tell if the minor type have the Y also or not. So I can't tell if those are male or female.
So okay, fair enough. And you can't tell anything about the sequence of time as to whether those three people touched or contributed at the same time, or who was first, who was last. You can't tell any of that. Is that correct?
You can't tell that for sure. Years ago, I read an article where they did steering wheel swaps, and they found that a lot of the times it was the person who drove it last, they removed the other people's and they left theirs, but not all the time. Sometimes it was variable. So yeah, you cannot definitively give any kind of order or time frame on those contributors.
So just taking your example, if I may, for a moment, of the steering wheel, I'll say maybe typically, I'll use that word, more than one person may drive a car. Would you agree?
Yes, I have 2 daughters, so they drive mine. Yeah.
Okay. So based on the mechanism of DNA transfer, it would be likely that all 3 of your daughters would have DNA on your steering wheel.
Yes, most likely I would expect it to be a mixture on my steering wheel of the 3 of us.
And let's say you drove the car 80% of the time, would that necessarily mean that perhaps 80% of the DNA would be yours?
Well, and that's what that study was kind of hypothesizing, that that's how it should work, but they found that it was highly variable. That's not necessarily how it works.
And in fact, they're your daughters, but one of them may be a more high contributor than others. That's not necessarily tied to genetics, is it?
Correct. They could be a better shedder, or maybe they just don't wash their hands as often. They could lose more DNA than I do, even if I drive it more. That's possible.
So with all of your expertise and experience, and as hard as you've worked on this case, you can't tell the jury whether Mr. Thompson ever touched that gun safe, can you?
No, I can only testify that the DNA is consistent with his being on there, but not how it got there.
Thank you. I have no further questions.
All right, anything else, Mr. Fedora?
Nothing further, Your Honor. Thank you.
You may step down, thank you. Later. Mr. Fedora?
Thank you, Your Honor. Thank you, Your Honor. With that, the people would offer into evidence, this is by stipulation, People's Exhibit 47, and this is Ms. Graham's it's Ms. Graham's lab report that's referenced in her testimony.
Mr. O'Farrell? Agreed, Judge. Exhibit 47 will be admitted.
And at this time, the people rest.
All right. Mr. O'Farrell?
Coach, Judge.
You may. Actually, we're going to break until next Monday. As I indicated earlier, we're not going to be a session tomorrow. And when you come back Monday, we're going to go into closing arguments and final remarks before you begin your deliberation. So once again, don't talk to anyone about the case, watch any news reports or listen to any news reports concerning it. Have a pleasant weekend, and we'll see you all next Monday at 9 a.m. Right? You can be seated. Record to reflect the jury is not present at this time. Anything for the record, Mr. Fedora?
No, Your Honor.
Mr. O'Farrell?
Your Honor, with the conclusion of proofs, I would move for a directed verdict of acquittal. Courts listened to the evidence in this case on 3 different occasions. And I know the court's well familiar with the standard. Unless the court wants further comment from me as by way of argument, I would defer to the court's decision.
All right. Any response, Mr. Fedora?
The court wants me to go through the elements. There's significant evidence here that the defendant was the individual who had shot and killed Mr. Parham. Considerable evidence of premeditation, testimony of former Detective Wenzel, who testified as to the fact that the defendant had been friends with an individual, Brandon Pennywell, who the defendant had suspected this is actually testimony through Lisa Curtis that she was aware, the defendant was aware that the individual, Antonio Parham, was suspected to have killed Lisa Curtis's son, Brandon Pennywell, and the defendant's good friend, Brandon Pennywell. So there's considerable motive there for the defendant to murder Mr. Parham. The facts are clearly beyond a reasonable doubt established. The defendant was the person who did so. The firearm was found to be the firearm used in the homicide was a gun that Ms. Curtis had given the defendant. Analysis of the photograph of the firearm, the type of firearm, etc., by Grell Rousseau established that casings that were found at the scene of the crime matched that firearm. The laser sight that was found at the scene of the crime matched that particular firearm. There was DNA extracted from that laser sight that was consistent with the defendant having handled the laser sight and placed it on the firearm. And then a witness who observed the
Mr. Fedora, you can
individual.
save your closing argument for the jury.
So, I mean, basically there's a plethora, significant amount of evidence there. Based on that, I just asked the court to deny the motion.
Well, as Mr. O'Farrell correctly stated, I've heard this case 3 times now. I'm intimately familiar with the facts of this case because they all were consistently the same during those 3 trials I've sat through. Taking the evidence in light most favorable to the non-moving party like I'm required to do, I do find it's a sufficient amount of evidence to allow the trier of fact to determine whether or not the defendant is guilty of these crimes he's charged with. and I will therefore deny the motion for directed verdict. Anything else, Mr. Fedora?
No, Your Honor.
Mr. O'Farrell?
No, Your Honor, thank you.
And also, I understand that both counsel had a chance to look at the finalized jury instructions. There were no objections by you, Mr. Fedora,
No, Your Honor.
or you, Mr. O'Farrell?
No, Your Honor.
Okay, we will